Hello, I welcome you all to today's session where we are going to discuss about the next group of biomolecules, that is that of the lipids. But before we discuss the lipids, we are left over with certain parts or portions of the other two biomolecules that we have already discussed, that is that of carbohydrates and that of proteins. So in previous sessions, I had told you that sugars exhibit various forms of isomerism, and the isomerism that are commonly marked in sugars. One already in the previous session I had told that it is D or L isomerism. I have already told about the aldose ketose isomerism. I have also told you about the pyranose and the furanose ring forms. I have also told you about the aldose and ketose isomerism, that already I have told. Then there are two more types of isomerism that are left with that we are going to discuss now. One is called as the alpha and beta anomerism. Let me change the color so that it becomes more prominent. In case of alpha and beta anomers, the ring structure of an aldose, you know that it is hemiacetal ring. And why it is called as hemiacetal? Because it is formed by a combination of an aldehyde and an alcohol group. Similarly, when we talk about the ring structure of a ketose sugar, it is called as hemiketal, as it is formed between a ketose group and an alcoholic group. Crystalline glucose, that is called as alpha D glucopyranose, and this cyclic structure is retained in the solution, but the isomerism occurs at position number 1, that is the carbonyl or anomeric carbon atom, and as a result the mixture of alpha glucopyranose will be having around 38% of D pyranose and 62% of you can say beta pyranose, and less than 0.3% is represented by alpha and beta anomers of glucofuranose. So here if I draw this particular ring, and at carbon atom number 1, if this is the orientation, then it is my alpha form, rest of the positions remaining the same. And if this is the orientation, then it is the beta anomeric form. I hope I have made this clear. Next, we will come to the next type of isomerism that are called as the epimers. So exactly what are epimers? Isomers which differ as a result of variations in the configuration of the hydroxyl and protons on the carbon atom number 2, 3 and 4 of glucose are known as epimers. So first let me draw the straight chain or the aliphatic form of glucose. This is the straight chain form of glucose. This is carbon atom 1, 2, 3, 4, 5 and 6th. If this second, third and fourth positions you consider about, then you will find the condition of the epimers. So first let us consider that how from glucose you are going to get mannose. Basically you change the orientation at this carbon atom number 2. So here these are the 6 chains of carbon atoms. This remains the same, only here I make the orientation of H and OH group different. At third carbon atom as such they are different. The rest of the carbon atoms retain the position. Now from glucose you have got mannose. I hope this is easy for you to understand. This is mannose. So it is about this carbon atom number 2 where orientation of this OH and H group is becoming just reverse, and that is epimerized at carbon atom 2. Another is that of galactose. So galactose is a epimer at carbon atom number 4. So here you have to remember that let us draw the straight chain first. This is H. This remains the same because we are drawing galactose, but mark this one. This is carbon atom number 4. Here is the OH and here is the H. At this position it is changing. Now rest of the things they remain the same, and H. So if this is the epimer of mannose and this is the epimer of galactose, then once they fold into the ring structure, then how will be their pyranose ring? So for glucose we know that this is carbon atom number 1, this is carbon atom number 2, this is carbon atom number 3, this is carbon atom number 4, this is carbon atom number 5, and this is carbon atom number 6. For mannose, because it is epimer due to its second carbon position, so first carbon will have HOH, but here the orientation will be opposite. Third will also be OH and H. Rest of the conditions will remain the same. So we have got the pyranose ring form of mannose, and for galactose the combination that we have to draw at carbon atom number 4. So at first orientation is same, second orientation same, third it is reverse, fourth it is reverse, this is fifth, and this is the sixth. So this is all about the epimers. Now let us come to another important term, that is reducing and non-reducing sugars. When I was teaching you about the disaccharides, I have many times told this term repeatedly. So some of you have already asked us doubts that exactly what do we mean by this reducing and non-reducing sugars. Sugars are classified as reducing or non-reducing depending upon the reducing properties attributed to the free aldehyde or the ketonic group of the sugars. Mind, it is due to the free aldehyde or keto group of sugars. The sugars which will be having a free aldehyde or ketonic group, they can reduce the cuprous ions which are present in copper sulfate to the cupric state. So they can change Cu2 plus into Cu plus state. This property is basis of Benedict states as well as Felling's test that we have done during our practicals, and that helps for the detection of or for the presence of, you can say, glucose or any other substance in the you can say human urine or in the food sample, etc. Benedict's test, we have understood that it is comparatively more common. The Benedict solution that contains blue colored alkaline solution of copper, that is the cupric sulfate, a reducing sugar changes it into insoluble reddish cuprous oxide. Already cupric sulfate is present Because of the reducing sugar, it is changing into the cuprous oxide state. On gentle heating, cuprous oxide separates as a precipitate, and the final precipitate may appear green, yellow or orange, or to brick red color depending upon the amount of reducing sugar. Once again I tell that the Benedict solution contains the alkaline solution of cupric sulfate, and once you are adding a reducing sugar to it, either present in the urine sample or in the food sample, it will be changing into the reddish color cuprous oxide, and that exactly is the basis of Benedict's or Felling's test. Once you are heating it gently, then the cuprous oxide separates as a precipitate, and the final precipitate may appear depending upon, if less amount of sugar is present, it can be yellowish. If moderate amount is present, it can be greenish, and if so comparatively higher level of sugar is present, it is orange. And if the sample is loaded with sugar like that of the fruit juices or plain sugar solution, in that case it will be brick red. I hope I have made this clear. Now let us move over to the part that we were left out for the structure of proteins. So for the structure of proteins, yesterday we had discussed about their coagulation and all, and today we will be continuing with classification of proteins. What are the different classes and how the proteins are grouped into different categories. So once we talk about classification of proteins, proteins are classified in several ways. The major types of proteins, they are classified generally based on their function. They are also classified based on their chemical nature and their solubility properties, and sometimes how they are nutritionally important to us. So first let us see that depending upon their function, how we have discussed or divided or classified them into different categories. So basically we are talking about the functional classification of proteins, right? So based on the functions they perform, then the proteins are classified into the following categories. So basically you know that proteins perform various functions in our body, and that is why they can be classified first obviously as structural. As the name indicates, they make various structures of your body. Definitely it will be, examples under this will be keratin that is present in your hairs or nails, or it will be the collagen protein that is present in the matrix of bones or cartilages. The second one obviously will be the enzymes. Most of the enzymes that are present in your body are proteins. Remember the protein digesting enzyme that is called as pepsin. During the respiratory cycle, we have studied about hexokinase enzyme during glycolysis. These all are proteinaceous enzymes. The third one will be the transport proteins. Definitely when we talk about the transport protein, the first thing that comes to our mind is hemoglobin, that is present in blood and helps in transport of carbon dioxide and water. Apart from that, we also have serum albumin. This helps to maintain osmotic pressure of blood plasma and helps in smooth functioning of it. Next one will be hormonal proteins. Hormonal proteins are actually hormones which are basically proteinaceous. And the best example under this, it will be insulin. And this is the most commonly studied hormone also. Your growth hormone also comes under this category. The next category will be the contractile proteins. The name indicates they help in contraction and relaxation of muscle fibers. And the actin and myosin and the sliding filament theory that we have studied, they will be coming under this category. So examples under this category will be actin or myosin proteins. the other next category will be that of the storage proteins. Now we have studied that storage carbohydrates, so they are quite common to us, but what about the storage proteins? And actually, are protein storage in nature? Yes, you remember ovalbumin. This is a protein which is present in yolk, and that provides food to the developing embryo. Another one under this category will be glutenin. These are storage proteins. Next category will be that of the genetic proteins. Genetic proteins, basically they help in folding of DNA. So genetic proteins will be having examples under them as histones. They help to fold the DNA so that it is accommodated within the nucleus. Next, there will be defense proteins. Obviously, the antibodies of our body. They will be the example under this category. Plus, here is another interesting example. The snake venom. That also contains a lot of proteins, and that is from the defense point of view of the snake. Then there will be receptor proteins. And receptor proteins, basically they are present on the cell surface of certain viruses, our cell membranes. When we had studied the mechanism of hormone action, we have come to study about this in details. So this is all about the different type of functional classifications of proteins. So if I try to sum it up, they can be structural proteins, they can be enzymatic or catalytic proteins, they can be hormonal proteins, they can be storage proteins, they can be contractile proteins, they can be transport proteins, they can be genetic proteins, or defense or receptor proteins. Proteins are also classified based on their chemical nature and solubility. So next category will be based on their chemical nature and solubility. So here, this is a more comprehensive and popular form of classification of proteins, and it is generally based on the composition of the amino acids, the structure, the shape, and obviously the solubility properties of the proteins. And depending on this, the proteins are classified into different categories, and this is a broad classification. Let me write it in this way. So the broad group is proteins. Their major classification will be into three main groups. They can be simple proteins, they can be conjugated proteins, and they can be derived proteins. So basically there are three types: simple, conjugated, and derived. Now let us discuss the categories one by one. The simple proteins, they contain only the amino acid residues, nothing else. The conjugated proteins, apart from amino acids, they will be containing a non-protein moiety, and that will be called as the prosthetic group. Prosthetic group is also called as conjugating group. Once we come across the enzymes, this property will be more clear to it. So here in conjugated proteins, there will be an amino acid part to it definitely. Otherwise, how it will be classified to be a protein? Plus it will be having a prosthetic group, which is a non-protein part that is attached to it. So that is why the name is conjugated. The main protein is conjugated or associated with a prosthetic group. And the third one, the derived proteins, they are the denatured or degraded proteins, which can be originally simple proteins or conjugated proteins. So if you denature or degrade the basic two types of proteins, then simple proteins or conjugated proteins, then you will get derived proteins. Now let us come to the category of simple proteins again. Depending upon their structure, simple proteins are classified into two main categories. They can be globular proteins or they can be fibrous proteins. Under this globular proteins, the structure or the name itself indicates that they are spherical or oval in shape. So if I sum up their characteristics, then they will be spherical or oval in their appearance. Secondly, they are quite soluble in water, because the polar groups that will be projecting towards the outer side. And the different categories under this, the first category will be the albumin. Already just now I have told that it is a serum protein. Albumin is quite soluble in water and dilute salt solutions, but it is coagulated by heat. The serum albumin, the ova albumin, lactalbumin present in milk, they are all categories under this. So albumin can be present in serum, can be present in the yolk of ovum, or it can also be present in the condition or in the soluble condition in milk. So it is serum albumin, this is ovum albumin, this is lactalbumin. Second category under this will be globulin. Globulin are quite soluble in neutral and dilute salt solutions. Example will be obviously serum globulins or antibodies, and the vitelin. This is a new term to it. Once you study the structure of Graafian follicle in details, you will come that it has a membrane which is called as the vitelin membrane around it, and this vitelin is a globulin protein that is present over there. Then the third one is glutenin. Storage proteins already I had told you. The glutenin is a storage protein. Glutenins are soluble in dilute acids and alkalis, and they are generally present in plants. Glutenin is present in wheat. Gluten intolerance, you might have heard about persons who are allergic or they show irritable bowel syndrome when they take bread or products related to that, or things which are made out of wheat flour. So in that case, this protein glutenin is present in wheat, and oryzanin, that is present in rice. So you have studied two examples. One is glutenin and oryzanin. Then the next category will be prolamins. This is a comparative new term to it. Prolamins are generally soluble in alcohol, that to 70% alcohol. There is a protein which is present in maize that is called as zeain. The scientific name of maize is Gia maize. So this protein zeain comes under the category of prolamins. The next one will be histones. Histones, many times previously we have discussed. They are associated, basic proteins associated with DNA, and they help in folding of DNA. They are strongly basic proteins and they are quite soluble in water and dilute acids, but in ammonium hydroxide they are insoluble. Next one is globins. This one was globulin, but here I am talking about globin. So globin is a protein which are generally considered along with histones, but globins are not basic proteins and they are not precipitated by ammonium hydroxide solution. That is the difference, but they remain associated with DNA. We will study about non-histone basic proteins. Globin is an example of that. It is a cylindrical structure and the solenoid form of DNA coils over it. Once we study about compaction of DNA in molecular basis of inheritance, this concept will be more clear to you. Next one will be protamins under this category. So protamins are strongly basic proteins and they resemble the histones, but they are smaller in size but quite soluble in ammonium hydroxide solution. They are also found in association with nucleic acid, and particularly they are called as the sperm proteins. So this is all about the globular proteins. Now let us come over to the category of fibrous proteins. So fibrous proteins are fiber-like obviously in shape, and they are insoluble in water. So they are fiber-like, insoluble in water. Globular ones are soluble, this one is insoluble, and they are resistant to digestion. Basically, globular proteins can be digested by your body, but these are not digestible. Fibrous ones are not digestible by your system. Albuminoids or scleroproteins constitute the most predominant group of fibrous proteins. These names might be sounding strange to you. Now let us come to the categories and you will become more clear to it. Collagens, many times I have already told. So collagens are the first category under this. They are connective tissue proteins, but tryptophan amino acid will be absent in them. They, on boiling with water or dilute acids, become gelatin, and that is soluble and digestible. I hope you have understood what I am hinting at. Ice creams, jellies, other things, they have a stabilizing substance to them. That frothy appearance when your ice cream melts at room temperature is because of the fact that the base substance milk is mixed with gelatin, and this gelatin is nothing but this is the collagen protein, which on boiling with hot water or dilute acids becomes that jelly like in its appearance and helps to emulsify your ice creams. The second category under this will be the elastins. When I was talking about muscle contraction, I have told you about this protein. These are the proteins which are found in tendons, and that helps the stretchability of tendons, or the property how tendons help to move the joints or muscles, that depends on it. And the second thing is that in arteries also, wall of arteries also, these elastins are present. So blood, when it is pumped within the artery with pressure, it helps to dilate the artery a little, not more, but to some extent. Then next one will be keratin. Keratin, many times since the previous sessions I have discussed. Keratin is your hair protein, and it is present in the exoskeletal structures. In hairs and nails, so many times I have told you, they are also present in the horn of horned animals, like in cows, in case of buffaloes, goats, sheep, in their horns it is present. Human hair keratin contains around 14% of the amino acid, sulfur containing amino acid cysteine. Now let us move over to this category, that is of conjugated proteins. So conjugated proteins, obviously they will be having a prosthetic group attached to them. So here you will be finding that nucleoproteins will be the first under this category. And nucleoproteins will be having nucleic acids associated as the prosthetic group. Example will be nucleohistones. See, already we have studied histone is a simple protein, and once you are associating it with a prosthetic group of nucleic acid, then it is becoming conjugated. The concept is not that difficult to understand. Second one is glycoprotein. The name itself indicates that the proteins have the prosthetic group of a carbohydrate. The glyco word is derived from the carbohydrate only. So when carbohydrate is attached to the protein, then it becomes glycoprotein. Example is mucoprotein, and that is mucin, which is present in your saliva. So example under this will be mucin of your saliva. The third category will be lipoprotein. The name itself indicates lipids here are present as the prosthetic group. So here the example will be serum lipoproteins. In membrane structure of plasma membrane, we have studied about lipoproteins. So lipoproteins are lipid plus protein. Definitely they are coming under the conjugated category. The next one is phosphoproteins. The name itself indicates phosphate group is present as the prosthetic group. So in phosphoproteins, the examples will be the vitelline, casein of milk. So this you have to remember. It is casein which is present as the milk protein. Remember that it is a conjugated phosphoprotein. And second one is obviously the vitelline. See, Vitelin once I had told you previously also. So whenever I had told you that, I have told as a structural one, but here I am telling you as its chemical nature. The next one is chromoproteins. The word chroma is associated with color. So definitely they will be associated with coloring substances. So prosthetic group is colored in nature, and most important example and most famous is hemoglobin under this category. Cytochromes, which we have studied in our electron transport system, they are also proteins under this category. And the last under this category but not the least, they are the metalloproteins. So metalloproteins you are able to remember because the prosthetic group is obviously a metal. Iron, copper, zinc, cobalt, manganese, etc. will be present. Carbonic anhydrase, when I was teaching you the respiratory system, we have studied about this enzyme very frequently. So this enzyme comes under the category of conjugated proteins. To sum up, the conjugated proteins can be nucleoproteins, glycoproteins, lipoproteins, phosphoproteins, chromoproteins, and metalloproteins. Now let us come to the third category that we have left over, that is the derived proteins. I hope the third category you are finding it over here. The derived proteins are of two types. They can be either the primary derived proteins or the secondary derived proteins. So the primary derived proteins are denatured or coagulated or fast hydrolyzed products of proteins. The secondary derived proteins are degraded, and they are due to the breakdown of the peptide bonds which are present in the products of proteins. So under the primary derived proteins, we will be getting coagulated proteins. So coagulated proteins are the denatured proteins which are produced by denatured, or they are the agents which denature them can be heat, can be acids, can be alkalis. They can be the proteins which result due to cooking, or they can be the proteins of egg white when it is boiled. So coagulated proteins are basically denatured by heat, acids, or alkalis. Under the example of this, there will be the cooked proteins of dal, chana, or rajma, or it will be egg white after it is hard boiled. Second one will be proteins. The spelling will be like this. Actually, we should call it as proteans. They are not the South Africans as we use in our other slang languages. These are the earliest product of protein hydrolysis by enzymes or dilute acids or alkalis. Remember, from a fibrinogen, fibrin is formed, and fibrin is insoluble in water. In clotting we have studied this. So when fibrinogen changes into fibrin, this fibrin is a protein, right? Then the third category under this, or I can say the other category under this, will be the metaproteins. This term I think you are a little bit more familiar with. So what are these metaproteins or superproteins? They are the second stage of products of protein hydrolysis which are obtained by treatment with slightly stronger acids or alkalis. Like a protein is present, whether it is a simple protein or conjugated protein, you put acid or alkali which is a little bit stronger in its strength, then whatever you get, they are the metaproteins. Now let us come to the second category, that is this secondary proteins. So let me write it over here. Secondary derived proteins, they are the progressive hydrolytic products of protein hydrolysis, particularly proteases, peptones, polypeptides, peptides which result during your digestion process. They are coming under this category. So the next one is that of the nutritional classification of proteins. So next classification of proteins that we are going to study, it is based on their nutritional value. In the beginning I told you that we will be classifying proteins depending on their various properties. So the next property that we are going to classify, that will be the nutritional classification. So the nutritional classification tells you that the nutritive value of proteins is determined by the composition of the essential amino acids. From the nutritional point of view, they can be complete proteins or first class proteins. So these proteins have all the 10 essential amino acids that are in that too in the required proportion, which is required by the human body to promote good growth of the body. Egg albumin, milk casein will be the examples under this category. The second one is that of the partially incomplete protein. These are the proteins which are partially lacking one or more essential amino acids and they can promote growth moderately. Example, proteins which are present in wheat or rice, because lysine, threonine, this kind of amino acids are absent in them. So they are coming under this category. Third one will be incomplete proteins. So incomplete proteins will be completely lacking one or more of the essential amino acids and they do not promote growth at all. Just like now I told gelatin lacks tryptophan, zein, it lacks tryptophan and lysine, and those kind of proteins if you are going to take, they are not going to help in the growth of your body in any way. So with this we have come to the end of this series of discussions where we were discussing about the different groups of proteins. Thank you.